All right, let's get started on the next chapter, chapter 13. This is going to be meiosis. Last one was mitosis. All right, again, here's your roadmap. So again, how does meiosis occur? And how do you compare it to mitosis? This is sort of the compare and contrast. How meiosis promotes genetic variation, independent assortment, crossing over, and then fertilization. We touched on these a little bit last time. What happens when things go wrong? Okay. And then what are the benefits of meiosis, right? Purifying selection hypothesis and changing environmental hypothesis. All right, so again, key definitions. Gonads are the organ that make the gametes, testes in males and ovaries in females. These are your gonads. These make your sex cells. Homologous chromosomes or homologs are a pair of chromosomes that contain the same type of gene. Now remember, we have 23 pairs of chromosomes, 46. So homologous chromosomes, let's say chromosome 3 from dad, well, chromosome 3 from mom are going to have the same genes on them. They're going to have the same information. They're homologous chromosomes, okay? Now we'll talk about alleles in a minute. There's some slight variation in those, but the genes that are being coded for are the same, right? So if your genes for height are on 3, your gene for height will both be on your dad's chromosome 3 and your mom's chromosome 3, okay? And genes, again, are a section of DNA that influences one or more hereditary traits of an individual. Genes are the region on the chromosomes that code for a particular messenger RNA, or they really code for a particular protein, right? The messenger RNA just transfers that information. Okay, alleles are different versions of a specific gene. That's what I talked about here. That's how you have this variation. The gene that codes for hair color is going to be the same gene. They both code for hair color, but they have some slight variations in them. Okay, but the gene still codes for the same product. So that's what alleles are. Remember, ploidy number is the number of chromosome sets a cell has. So we are diploid. We have 2 sets, one from mom and one from dad. If you're haploid, you only have 1 set of chromosomes, and they're notated. You'll see this 1N or N, that's haploid, or 2N, that is diploid. Okay. Haploid cells are going to be your gametes, right? And then somatic cells, all the other cells are going to be your diploid cells. Excuse me, I still haven't gotten over this bronchitis. So during sexual reproduction, egg and a sperm, right, they unite to form a new individual, and this process is called fertilization, right? Meiosis is that nuclear division that precedes the formation of gametes, eggs and sperm, because you have to get rid of half of the chromosomes. So resulting in halving of the chromosome number, right, or having the ploidy number. Is usually accompanied by cytokinesis. So you're going to split again, that's cell division. So division of the cytoplasm into 2 distinct cells, right? Remember, this is a special type of division because when fertilization occurs, you're going to add those chromosomes in, chromosomes from the sperm and the chromosomes from the egg. If you haven't gotten rid of half of them, you're going to have too many chromosomes. We'll see later on, too many is not good. Okay? So if you compare and contrast the two, mitosis is cell division used for asexual reproduction, growth, repair, right? All the genes come from one parent, right? The cell just splits into 2. Mitosis, cell division that leads to the production of all other cell types, right? Referred to as your somatic cells. And the genetic material is copied and then divided equally. So you copy the DNA, and each new cell gets 1 copy of that DNA. So daughter cells are genetically identical to the parent cells. They're clones of each other. Meiosis is sexual reproduction, right? Used to make the egg and the sperm. In meiosis, cell division that creates these gametes results in halving of the chromosome number. They go from diploid to haploid. So the daughter cells have half the amount of genetic material as the parent cell. And again, occurs in the gonads of sexually reproducing organisms. Now remember, the cells in their gonads, like the testes and ovaries, they're going to go through mitosis too until they're going to become either a sperm or an egg. Then they go through meiosis. So every organism has a characteristic set of chromosomes, that chromosome number, right? So research by Stevens in '06 indicated that the fruit fly has 8 chromosomes. Okay, she was the first to study mitotic chromosomes. Meiotic, not mitotic. I'm sorry, meiotic chromosomes. Okay. Sex chromosomes determine the sex of an individual. Those are two. We each have one. We each have 2 sex chromosomes, right? So in many animals, females have 2 X chromosomes, XX, and males have an X and a Y. So the Y is the one that determines the sex of the organism. Autosomes are non-sex chromosomes. So that's all the other 44 chromosomes are going to be autosomes. Okay. Again, this just shows the pairs. So here you've got types of chromosomes that often come in pairs. So here you've got the 2 3s, 2 chromosome number 3, 2 chromosome number 4s, 2 chromosome number 2s, right? So these are about the same in the male and the female, right? Coding for the same information. Males are going to have a Y chromosome and 1 X chromosome, whereas females are going to have 2 X chromosomes. And again, this just shows you that it's sort of a karyotype, but here are the, let me shrink this down so you all can see it. Here are the homologous chromosomes. Here you have, instead of number, they're A, B, C, D, E, F, G, H, I, J, K. X is going to be a sex chromosome, right? What they're showing here is these 2 right here, these 2 C chromosomes, one from dad and one from mom, are homologous chromosomes. You get 1 C from dad, you get 1 C from mom, you get 1 A from dad, you get 1 A from mom. So they have the same, they code for the same genes. Now there may be variations in those genes, but they code for the same genes, right? No. So again, here's the, again, homologous chromosomes, the gene for eye formation. Here's an allele for round or shaped eyes. Okay, so this gene right here on both these chromosomes, because these are homologous chromosomes, this area here, this gene codes for eye formation. Okay, on both of them, but there's a slight variation. In this case, this gene is for more narrow shaped eyes. They still code for eye shape. They are just slight variations on the allele. Okay. Now, during definitions unique to mitosis here, this is actually going to be, I don't know why it says mitosis, it should be meiosis, is chromosome replication, synapsis, and crossing over. So all this whole thing with sexual reproduction, if you remember, is ultimately about genetic variation, right? Genetic diversity. You get half your chromosomes from mom, you get half from dad. That's a lot of genetic diversity. But also when you're making your sex cells and dividing in meiosis, you also have some other mechanisms for diversity. In this case, one of them is called crossing over. So in the first part, and we'll talk about this later, the first part of meiosis 1, we'll call it, you have your 2 homologous pairs, right? They're sister chromatids. They've been replicated during interphase, okay? So during this S part of interphase, so here are your homologous chromosomes and the sister chromatids. So they're going to come together and they're going to form this synapse. They're going to synapse during prophase 1. We'll talk about prophase 1 a little bit. But they couldn't synapse. But remember, since the information is this, they're coding for the same genes, the genes are the same, you can get this crossing over. You can swap a little piece of this chromosome from mom with this chromosome from dad. Okay? So crossing over usually occurs at least once in each non-sister chromatid. Right? So the synapses, homologous chromosomes pair together in preparation for crossing over, and crossing over produces new combination of alleles on the same chromosome, right? So this happens during prophase 1, and we'll, like I said, we'll go over that in a minute. But so now when these split and go in different cells, the sister chromatid, right, there's going to be some more genetic variation, and it's just another way to increase that genetic diversity. All right, again, ploidy number. Cells ploidy number. N is 1N, that's haploid. 2N, you can be tetraploidy, not you, but actually that's bad when that happens with some of them, but some plants and things are polyploidy and tetraploidy, right? Indicates the number of each type of chromosome present. Haploid, again, organisms whose cells contain just one of each type. Diploid, whose cells contain 2 versions of each type of the chromosome. In us, diploid cells have 1 parental chromosome and 1 from dad, and 1 maternal, 1 from mom. One each. Polyploidy, 3N, 4N, etc. Organisms with 3 or more versions of each type of chromosome. You see these in plants and some other organisms like that. So we're not going to worry about those there. You can maybe talk about those next semester if you take the second half. But if you look at haploid chromosomes and diploid, we have 23 pairs, right? Or 46 total. Okay. So you got to realize the number of just having more chromosomes does not mean you're more higher up in the evolutionary tree per se, right? Because dogs have 36 pairs and 72 chromosomes. So if the more was made them better, we'd all be run by dogs. Some of us are. Fruit fly only has 4 and 8 total. Chimpanzees, 24, 48. The bullet ant has 1 chromosome pair and 2 total. Garden peas and then corn or maize has 10 to 20. So the number of chromosomes has nothing to do with how either evolved or whatever you want to, however you want to put it, of the organism. Okay. So again, this is just one of those vocabulary charts, a good summary table if you want to look over, right? If you have questions on this, let me know. All right. So meiosis, just before meiosis begins, each chromosome in the diploid parent cell is replicated, right? So again, you're going to copy your information first. When replication is finished, each chromosome has 2 identical sister chromatids, right? That's where you get those little X-shaped things we talk about, because you copied them. 2 sister chromatids, but it's still considered 1 chromosome as long as they're connected. Okay, so they remain attached during most of meiosis. Okay, what they're saying is these 2 remain attached during most of meiosis. The 2 attached sister chromatids are still considered a single replicated chromosome. I just talked about that. All right, here we got meiosis, going separate ways. They call it here. So cells that carefully separate homologous pairs of chromosomes so the gametes receive 1 of each type. So let's say there's a bullet ant. It's got 1 chromosome, right? 1 pair, 1 from mom and 1 from dad. And you're going to get replication now before you're going to make these sex cells. So you replicate, right? You replicate mom's, the maternal chromosome, and you replicate the paternal chromosome, right? So 2 sister chromatid, 1 chromosome. You still only have how many pairs? You still only have 1 homologous pair at this point. All right. So meiosis consists of 2 cell divisions versus just 1 in mitosis. You've got meiosis 1 and meiosis 2. Okay? And the cell divisions occur consecutively, but they differ in what's going on. The purpose of meiosis 1 is to separate the homologous pairs, to separate the chromosomes you get from mom from the chromosomes you get from dad. It's random. Not all mom go into one cell and all dad go in the other cell, but you're separating the homologous pair. This is where you go from being, the cells go from being diploid to haploid. Meiosis 2 now is to separate the sister chromatid, a lot like you saw in mitosis. So the purpose of meiosis 1 is to separate the pairs of homologous chromosomes. So the homologs in each chromosome pair are separated and go into different daughter cells. Okay, you've got prophase 1, metaphase 1, anaphase 1, and then telophase 1. This is all part of meiosis 1. So during meiosis 1, like I said, the diploid parent cells produce 2 haploid daughter cells. You reduced or decreased the ploidy number. That is the purpose of meiosis 1, to have the ploidy number. Excuse me. The homologs in each chromosome pair separate and go into different daughter cells. And although the daughter cells are haploid, just N or 1N, each chromosome still consists of 2 identical sister chromatid. Remember, you replicated them, so you still have this 1 chromosome that is made up of 2 sister chromatid. Okay? So let's walk through it. Here's that bullet ant, I guess we want to call it here. You've replicated it. You've got the homologous pairs here. You go through meiosis 1. So right now, this is 2N. During meiosis 1, you have this after the cell division. After meiosis 1, you separate the homologs. So now you've got One cell here and one cell here, and these have gone from 2N to 1N because the daughter cells contain just one of the homologs, one of the homologous pairs. During meiosis II, now you're going to separate the sister chromatids so that at the end of meiosis II, or at the end of meiosis completely, you should have 4 daughter cells that each contain 1 chromosome each. Okay? And these are the ones that become the sperm and the egg later on, depending if you're male or female. All right. The outcome of meiosis is the reduction in chromosome number. And for this reason, meiosis is known as the reduction division. Okay? In most plants and animals, right, the original cell is diploid and the 4 daughter cells are haploid. It depends. We talked about some plants and some other ones, but for the majority of them, you go from being diploid to haploid. In animals, these daughter cells become the gametes. So this process, the whole process, is called gametogenesis. Genesis is to make, right? Create, and gametes are the gametes. So you're making the gametes. All right, so fertilization now results in a diploid zygote. So when 2 haploid gametes fuse during fertilization, a full complement of chromosomes is restored. You are restoring the ploidy number. So the cell that results from fertilization is a diploid cell now, because you have a haploid sperm and a haploid egg. They fuse together, they add their chromosomes together. So now they go from being 2 1N or haploid cells to a single diploid zygote. Right? So in this way, each diploid individual receives haploid chromosomes set from both mother and father. This is that genetic diversity that you get from sexual reproduction. So homologs are referred to as either being maternal chromosomes, the ones from your mom, or paternal ones from dad. And here you've got right here, here's the female gamete, 1N. Here's the egg, the male gamete, the sperm, which is 1N. You get fertilization, and the diploid offspring contain homologous pairs. So now you've gone back up to 2N. The zygote is now 2N. Now, going back, if you hadn't separated the homologous pairs, right, or something, you would have extra chromosomes in here now. That would be bad. We'll look at that at the end. So chromosomes condense. This is going to be prophase 1. Okay, chromosomes condense. A lot of this kind of looks like mitosis, but you got to remember what's really going on. The nuclear envelope breaks down. Remember, you've already replicated your chromosomes, so you have sister chromatids, right? The spindle apparatus forms and attaches to the chromosomes. Now the homologous chromosomes find each other and pair up. So you get chromosome 1 from mom pairs with chromosome 1 from dad. Chromosome 2 from mom pairs with chromosome 2 from dad. Chromosome 3 from mom pairs with chromosome 3 from dad. These lined up in pairs across the equator. Remember in mitosis, they were single file. Here, the homologous pairs, like kids, the buddy system, line up in pairs. The homologous pairs line up. Okay? So metaphase 1, the homologous pairs of chromosomes line up in the middle of the cell now. Anaphase 1, the homologous chromosomes are separated from each other. And one from each pair goes into the opposite poles of the cell. So they're pulling apart, okay? But in this case, they're still connected. The sister chromatids are still connected. So what you've done, you look down here, they line up, they pair up, line up across the middle. Now the homologous pairs separate and get pulled towards the poles. And then telophase 1, the nuclear envelope, starts to form, creating 2 nuclei again. So these nuclei are now haploid because they only have 1 of each type of chromosome. Spindle fibers break down. You get that invagination of the membrane during cytokinesis, and you form 2 haploid cells at the end of cytokinesis. So after meiosis 1, okay, both cells proceed directly to meiosis 2. You don't go back through interphase, because if you went back through interphase, you would copy your DNA again, and that would defeat the purpose. These go right from meiosis 1 into meiosis 2. Okay? So at the end of telophase 1 at cytokinesis, haploid cells contain chromosomes that each consist of 2 sister chromatids. These are still connected. And it is random. That the assortment is random. Which one goes into which cell is just completely random. So the result of meiosis 1, right, is chromosomes from each homologous pair is distributed to different daughter cells. They go into different ones. And meiosis 1 produces 2 cells that are haploid. Okay. Now the daughter cells after meiosis 1 are haploid, but remember the chromosomes, the sister chromatids are still connected, the ones you copied before. So now we'll jump into meiosis 2, and the job of meiosis 2 is to separate the sister chromatids. This looks a lot more like mitosis. So sister chromatids from each chromosome separate and go into different daughter cells, and you have the same steps: prophase 2, metaphase 2, anaphase 2, telophase 2, and then finally cytokinesis again. So spindle fiber forms from each cell and attaches to the chromosomes, right? Because you haven't got rid of all that nuclear envelope and everything else that goes away, right? The nuclear envelope and membranes break down again because you've got to get access to it. So here's meiosis 1. You have these 2 daughter cells that are haploid, but the sister chromatid are still connected, right? So now you jump right into meiosis 2. Meiosis 2, prophase 2, the nuclear envelope still breaks down and spindle fibers start to arrange. And now you go to metaphase 2. Chromosomes are lined up in the middle of the haploid cell. Again, these are going to line up in single file again. Okay, so here's prophase. Now you're red meiosis. Metaphase 2, they line up single file again. Right here. And then you go to anaphase 2. The sister chromatids separate and move to opposite sides of the cell. Now, again, this looks a lot like mitosis, right? But you know this isn't mitosis because you can see where you had crossing over, and that only happens in meiosis. Okay, so you separate the sister chromatids now. They go to opposite poles. And then telophase 2, again, you start to reform the nuclear envelope. Spindle fibers break down. The membrane starts to, you get the cleavage furrow coming in. Membrane starts to invaginate in. And then you go through cytokinesis next. And you end up with what? 4 haploid cells. These 4 cells now, depending if you're male or female, develop in either eggs or sperm. Again, meiosis 1, the resulting is what? 2 daughter cells that are haploid, right? Homologous pairs separate. After meiosis 2, the 4 daughter cells contain 1 chromosome each, so they are haploid still. And you have separated the sister chromatids, right? So these become the sperm and the egg. And again, this just shows fertilization does what? That's why I usually ask a question. And the question is, it forms the zygote, which now has restored the ploidy number. So you look at here you got, I guess this is Fifi right here. And this is haploid. Diploid is going to be 2 in right here. So here's going to be what? This is coming from Fifi, so the diploid egg goes through meiosis, the cell becomes an egg. And then you've got the dog from the fence jumping dog from up the street, jumps the fence, comes down, and it has haploid gametes, sperm and an egg. You have fertilization. Now you restore the ploidy number. The zygote now is to 2 in. And now it'll start going through several different thousands and thousands of mitotic divisions and cell differentiation later on to eventually become an adult or eventually an adult animal, right? Now you go the first few divisions when these go through it, there's no what we call cell differentiation. The cells are just dividing and making copies of themselves. Later on, they start to differentiate. That's how you get identical twins. If you have, I'm gonna go back. I guess I can do it real quick since I jumped into it. If the egg separates, okay, if the egg before it starts to differentiate, and it's just making copies of itself, right? It's just growing. But if those two happen to come apart, as long as they haven't started to differentiate, they'll still keep dividing. And that's the exact same information, right? That's how you get identical twins. Before this cell starts to differentiate, the zygote will pop into 2 different cells, or it could be 4 and 4. Fraternal twins are when you get two eggs get fertilized. That's why they're not identical twins. So that's the difference between the two. All right, so where does crossing over occur? We already went over this, right? It happens during when? It happens during meiosis 1. Okay? So replication here, you have the sister chromatids. The homologous pairs now, once they've been copied, are going to synapse during prophase 1. Remember that. And then crossing over is also during prophase 1, and then they're going to separate across. They'll separate it after they've crossed over. So again, key events in prophase 1, you look at here. Here you've got the homologous pairs, you've got the kinetochores, you get condensation, the sister chromatid. Homologous pairs get condensation, they pair up. You get a break in the chromatid DNA. You get that synapse, that's that bivalent formation, two of these together. You've got this cut here in the DNA in the chromosome. You get crossing over, and then you get this, start to get this partial separation of the homologous pairs, and then eventually they'll separate and pull apart. And you will have switched apart that little piece of DNA. And it can be, it could be down here too. It could be pieces all over. So now, what I wanted to talk about, the consequences of meiosis, the independent shuffling. Remember I talked about that? The homologous pairs, not all of mom go into one cell and all of dad go into other cells. It's called independent assortment, independent shuffling of the maternal and paternal chromosomes. Okay, that's diversity. You also get crossing over during meiosis 1, which results in 4 gametes with a chromosome composition different from any of the parent ones. Because the crossing over now has even changed these chromosomes so they're different from the ones that came from the parents. So you really can increase the genetic diversity here. So the change in the chromosomes produced by meiosis and fertilization are significant because the chromosomes contain the cell's hereditary material, right? All that information you're inheriting from your parents. Okay, next. So again, sexual reproduction leads to greater genetic variation. Bacteria and some other cells that go through asexual, some fungi, some other protozoans and things, they don't go through sexual reproduction, right? So they just clone themselves. So there's no genetic variation there. The only way you get that genetic variation is through mutations. So offspring produced from asexual reproduction versus sexual reproduction. Genetic variation in sexual reproduction results from the independent assortment of the homologous pairs and the crossing over. Way more genetic diversity through sexual reproduction than asexual reproduction. So again, independent assortment is the separation and distribution of the homologous chromosomes during meiosis 1 can result in a variety of combinations of maternal and paternal chromosomes. We have 23 pairs, right? So you can get 20, all those different ones can go into, get 10 from mom, and I'm going to have to do some math here. 10 from mom in this side, and then that's going to be 13 from dad on this side, and it'll be the opposite on the other cell. See, that's that massive amount of variation. Good, and it's completely random how they line up, how they pair up, which side of the pair they pair up to when they're pairing up. So each daughter cell gets a random assortment of maternal and paternal chromosomes, and thus a random assortment of those gene alleles, which generates a great deal of genetic diversity. And then you throw in crossing over and you think how much diversity there is. So again, here's 2. Here are the 2. Here we just got 2. This is going to be diploid. It's still diploid because it's got 2 sets, but it's only got 2 pairs of chromosomes. Right? Here's an allele that contributes to brown eyes and green eyes, and this one's going to be black hair and it's going to be red hair. During meiosis 1, the tetrads line up 2 different ways before offspring separate. They can line up this way, right? Or they can line up this way, okay? And when it goes through this separation now, you notice how these 2 are on opposite sides of each other, because it's random. So when these separate now, this one now you're going to have brown hair and black eyes. This one's going to have green eyes and red hair. This one over here now is going to have brown eyes and red hair. And this one over here will have green eyes and black hair. So different ways this individual assortment, random assortment of homologous pairs going into different daughter cells. Okay? So again, this probably should have put this slide before. This just again shows the alleles. This gene right here codes for eye color, but they are different alleles because they're heterozygous. We'll talk about that in the next chapter. Because this allele contributes to brown eyes, whereas this one contributes to green eyes. Okay, they still code for eye color, but they're different variations. Over here, this gene here still codes for hair color, but there are different variations in there. So that's what's called an allele. Okay. Okay, so differences, the key difference between mitosis and meiosis is the difference between the two processes is that homologous pairs in meiosis, right, but do not in mitosis, right? So meiosis produces 4 daughter cells that have half the genetic material. You have the ploidy number, you decrease the ploidy number, you reduce the ploidy number, right? You go from being diploid to haploid, those cells. Mitosis produces 2 daughter cells that are clones. They're genetically identical to the parent cell. So again, mitosis, you get 2 daughter cells that are diploid. Exactly the same. Meiosis, you get 4 daughter cells that are haploid. Okay? And you assorted these different homologous pairs into different cells. So again, now let's look what happens when you get some mistakes in here, and this can happen. Okay? Mistakes in meiosis and what we call chromosomal disorders. Okay? This is called non-disjunction. So chromosomes that fail to separate correctly undergo what they call non-disjunction. So non-disjunction results leads to the creation of gametes that have the wrong number of chromosomes. This is called aneuploidy. Remember I told you, give me one quick sec here. That was odd. All right. Aneuploidy. Remember I said having too many or too few chromosomes is not good. Okay. So here in meiosis 1 starts normally, but you get non-disjunction. So during meiosis 1, you fail to separate these 2 homologous pairs. So now the cells go through cytokinesis. Now they go through meiosis 2. They separate the sister chromatids. And now you end up with what? This one had an extra chromosome in it. This was missing one. So now the offspring, right, this is going to have an extra chromosome in it. So this is going to be N+1. And this one will be N+1. That's not good. And this one down here is missing a chromosome. So now it's going to be N-1 and N-1. Both of these are bad. Okay. So let's look at when non-disjunction takes place. Okay, because if it takes place earlier on, it's more detrimental than it takes place later on. So here during meiosis 1, if you have non-disjunction, failure to separate the homologous pairs, we looked at that one. So now this daughter cell is missing a chromosome, and this daughter cell has an extra one. Now it goes through meiosis 2. You separate the sister chromatids, and again you get this one N+1, N+1, N-1, N-1. All of these are going to be bad. All of the gametes now are going to be aneuploids. Okay, and this is bad. If you separate the homologous pairs during meiosis 1 successfully, and you undergo non-disjunction in meiosis 2, and you don't separate the sister chromatid, so you look over here, this one was fine because this one didn't go through non-disjunction, only this one did. So now you end up with N+1 and N-1, okay, because this has that extra chromatid. You didn't separate the homologous pairs. This is missing one. So those are bad. Usually these are going to won't survive, or if they do get fertilized, they don't survive. But in this case, the other 2 daughter cells are going to be okay. Okay, so it depends on when non-disjunction happens. So let's look at types of non-disjunction. These are these chromosomal disorders we're talking about. So trisomy is 2 gametes will have an extra copy of a chromosome and 2 gametes will lack a chromosome. So trisomy's got the extra and monosomy is missing one. Probably the best well-known example of trisomy is Down syndrome. Okay, and Down syndrome, the results from 3 copies of chromosome 21. Okay, so and that's the most common one in there. These children actually survive now into adulthood, and you've got actors and actresses and I think even a model was on the cover of one of the magazines. So Down syndrome, people can actually survive and have full lives now. That wasn't the case 30 years ago. But now it's actually, they live kind of, like I said, full, long, rich lives. Mistakes in meiosis are the leading cause of spontaneous abortions or miscarriages in humans. That's what I talked about. Usually, these are going to be what we call embryonically lethal when you have these changes in chromosomal numbers. Now you look at mother age in years, and this is the incidence of Down syndrome per birth. And this happens as the mother gets older. It's 1 in 1,000 here at their 30s. So below that, it's much even less. 36 or so, it's about 1 in 300. Okay. And then it starts to increase and increase. So up into 48, the chances are 1 in 12. It doesn't mean you're going to have it when you have children. In fact, a lot of women there are a lot more women having children up in this age, and you don't see as many cases of Down syndrome as you would think you would see. They used to think it was only the age of the eggs, the woman, but it turns out that you also start having chromosomal damage in males as we get older as well. And so this is that karyotype, trisomy 21. Trisomy means there's 3. In this case, it's going to be chromosome 21. You see in the karyotype, there's 3 chromosome 21s. Now this is an XX, so this would have been a female, right? Another, and again, like I talked about with Down syndrome, with all of these chromosomal abnormalities, you have various degrees of severity. Some people are really affected, other ones are not affected as much. Another example of trisomy is going to be Klinefelter syndrome. Okay, so have male sex organs but are sterile and may have female characteristics. Okay, this is going to be a male with an extra X chromosome. Okay, these are sex related, not somatic. So they don't make as much of the male hormone testosterone, so they tend to become what we call feminized, right? They can get breast development, small testicular size, female pubic hair pattern versus male, somewhat wider hips, longer arms and legs, narrow shoulders, again, poor facial growth. Frontal baldness is absent, but these all vary in severity. Some Klinefelter syndromes you can barely tell. Okay, this one is actually treatable if they can detect it early, especially before you go through puberty. They can start giving testosterone replacement therapy. So this one actually has a treat, right? They may have less facial hair, body hair, maybe less muscular than other boys. They don't have as much testosterone. But again, there are varying degrees of this, and this one is actually treatable. Again, down here, here's the karyotype of Klinefelter syndrome. You look at it and you see these 2 X chromosomes. Trisomy X, which is triple X here, so a female with an extra X chromosome. And again, these also vary in severity. So tall stature, sometimes they have that microcephaly, the small head. Vertical skin flows that cover the inner corners of the eyes. Okay, delayed development of certain motor skills, speech and language. But again, this is variable. Okay, learning disabilities, dyslexia. But again, there are varying degrees of this. Okay, weak muscle tone. And again, you look at the karyotype here and you see it down here. So this is going to be trisomy X, right? Because you see 3 X chromosomes in this particular karyotype. Turner syndrome is an interesting one. This is monosomy X, or Turner syndrome, which is XO. Okay, produces a phenotypic but immature females. Okay, So genes affect or involved in growth and sexual development. So you're missing one female chromosome. They tend to be, and again, these vary in severity. Some is very noticeable, other ones it's not as noticeable. Okay, short stature, failure to begin puberty, they don't make estrogen, infertility because they don't go through puberty, they have heart defects, certain learning disabilities. But again, these are all variable. There's another one that you can actually treat at puberty with estrogen therapy, right? There's actually a very good Law and Order SVU episode that deals with Turner syndrome. That where it raises a bunch of interesting ethical questions in it. So this particular one, the girl looks like she's I think 18, but she looks like she's say 12 or 11, prepubescent. And she ends up wanting to go off and marry this guy who's older. And so the question is, I mean, she's 18, right? So she's an adult. So can they stop her from marrying this guy? Secondly, the real question is, you can see why she wants to be normal. She wants to be normal, have a family and everything else, and not look like a little girl her whole life. And even then, even with estrogen replacement therapy, it's still, they probably have a shorter fertility window. But the bigger question is why does this individual like her? That leads to the ethical question: is he like her because he's really a pedophile, or is he something else? It's just a very interesting Law and Order. I wish we could watch it sometime. If you ever find it, it's really interesting to watch. It's a lot of interesting questions. Should you give them hormone replacement therapy? Whose decision is it? Because they're not adults when this would start. So really good. But again, there's varying degrees of this. Some people with monosomy Turner syndrome actually can go through puberty and you really can't tell that much. It just kind of depends on the individual. And again, you have the 1X chromosome here. All right, this again is that vocabulary. It's a lot of it's the same as the other one. Just really good study guides to look through and review these, right? Make sure you know what non-sister chromatids. I didn't go over this that much. Just remember, non-sister chromatids are chromatids belonging to homologous chromosomes. These are homologous pairs, so they all code for the same thing. So this is non-sister chromatid to this one, right? All right, again, this is just the difference between the two. And that's it for this, for meiosis, everybody.